Hello again. So we just finished glycolysis. That's part of aerobic cellular respiration where a glucose molecule or some other nutrient is partially oxidized and we make a little bit of ATP. We make some NADH. NAD plus is reduced to NADH. That's the dump truck filled up with electrons, high energy electrons, right? And we make 2 pyruvate molecules. Now we have to find out what happens to these pyruvate molecules as they move through the rest of aerobic cellular respiration. So I'm going to go back to our PowerPoint slides and we'll start on slide 19. That's how far we've covered from this for this chapter. So here we are at pyruvate oxidation and the citric acid cycle, or sometimes called the Krebs cycle. I love the little meme that I found online. Any case, we're going to focus on just introducing what the citric acid cycle is, and we'll take a look at it in this video. So here's some introductory information. I think I better get my pen going. All right, I think we'll be good. So in pyruvate oxidation and the citric acid cycle, pyruvate molecules are completely oxidized, and what's released are waste products. So pyruvate molecules are oxidized to carbon dioxide. So we can write that in. You can write in pyruvate molecules, and I'll get it started, but I won't have enough room here. Pyruvate is oxidized and carbon dioxide is released. That's the waste product of cellular respiration. That's what we breathe out when we respire as well, right? In this process, many coenzymes or electron carriers are made. Those are NADH molecules. So basically, we make a lot of these little dump trucks with high energy electrons in them. So many, and you can write this down, many coenzymes are made, or you can put electron carriers. In parentheses, I want you to write NADH and another molecule called FADH2. So I'm going to write down NADH, lots of NADH molecules made, and another electron shuttler called FADH2. Okay? These molecules are later going to be used to make ATP, but not in this part of the process. The third important introductory information bit is that this all is happening in the matrix of the mitochondria, in the space in the middle of the mitochondria. Remember that a mitochondrion has 2 membranes, an outer membrane and an inner membrane, and inside the middle is called the matrix of the mitochondrion. That's down here. That's where we are, right? All right, and finally, this is a cyclical process. The citric acid cycle is cyclical. That means that we start with one molecule and we end with it at the end of the cycle. So let's take a look at our kind of introductory slide, and then we'll go through the processes. All right, this is the slide that you should memorize, right? This is the process of aerobic cellular respiration, all mapped out for us. And so far, we've done glycolysis, right? In glycolysis, glucose is partially oxidized to pyruvate. That's all happening out in the cytosol. We make a little bit of ATP, yay, and some NADH. In the pyruvate oxidation step, pyruvate is converted into this molecule called acetyl-CoA. This is happening inside the matrix of the mitochondrion. This is kind of a prep step. What this does is it kind of prepares pyruvate to enter the citric acid cycle. And then there are a series of reactions in the citric acid cycle. And during that process, it looks like we make some more ATP by substrate level phosphorylation, and we make a lot of these electron carriers or shuttlers of NADH and FADH2. And it looks like they're going to be used later in oxidative phosphorylation to make a lot of ATP. Okay, let's get started. Let's look first at this step called pyruvate oxidation. So when pyruvate is made out here in the cytosol, it has to move into the matrix of the mitochondrion for pyruvate oxidation to happen. Here's a slide of pyruvate oxidation, and it looks a little intimidating. Let's look at it a little bit. So on the left, we've got our pyruvate molecule out in the cytosol. It has to move into the matrix of the mitochondria through a transport protein. So it moves in, and then this reaction happens, and this is a complicated reaction. It involves, hey look, it involves a dehydrogenase enzyme. Actually, it's called pyruvate dehydrogenase complex. This is a complex of lots of enzymes that work together to complete this reaction. What's going on in this reaction? Well, pyruvate is being converted to acetyl-CoA, whatever that is, right? There's lots of things happening. It's a multi-step process. So let's look at the steps that they've given us. The first thing that seems to happen is that carbon dioxide, and then you can see it here in gray, carbon dioxide from this pyruvate molecule is removed from the pyruvate molecule. This is kind of a waste product, right? And so we would say that pyruvate is decarboxylated. Its carbon dioxide is being removed. There's a waste product. What's left over then is oxidized. I know this is a redox reaction because I see NAD plus being reduced to NADH. Pyruvate is oxidized, and we really can't see that in this step. We don't see how hydrogens and electrons are being transferred around, but it's happening. So in part 2, what's left of pyruvate is oxidized. It loses electrons, and NAD plus picks up electrons and is reduced to NADH. So we've made more NADH. That's great. The third thing that happens is this molecule, what's left, this 2-carbon fragment that's left over, picks up a coenzyme. Coenzyme A becomes attached to the 2-carbon fragment, and it comes attached here with at a sulfur group. That attachment is pretty weak. Coenzyme A is not going to stay on our molecule for very long. But we've got acetyl-CoA. This molecule now is ready to enter the citric acid cycle. I think of this as the prep step. But kind of important things happened. We released some waste product, carbon dioxide. We've seen another redox reaction where pyruvate is oxidized and NAD plus is reduced to NADH. So we've made more of those filled up dump trucks, if you will, those high energy electron carriers. All right, let's see what happens to acetyl-CoA as it moves through the citric acid cycle. Remember what we're trying to do here is, or what's happening here is, pyruvate is being completely oxidized. In other words, we're tapping into all the energy in pyruvate and using it to make lots of NADH and something called FADH2. Okay, this is probably the scariest diagram in our textbook, but don't fear, you don't have to memorize it all. I put some things kind of on the outside of the diagram that are important things to remember about the citric acid cycle. Let's look at what's happening, and we'll try to fill in the blanks on these starred boxes. So up at the very top, we've got, oh yes, pyruvate oxidation step has happened. So pyruvate gets converted into acetyl-CoA. Acetyl-CoA is this kind of unstable molecule. And what happens then is we've got this circle of reactions, don't we? There are, looks like 8 different reactions that happen, and we start with this molecule, acetyl-CoA, interacting with oxaloacetate. But by the end of the citric acid cycle, we end up with oxaloacetate again. So in step 1, it looks like acetyl-CoA enters the cycle by reacting with oxaloacetate. So you're going to want to fill in, that's never going to fit oxaloacetate. Okay? Each one of these steps is going to require an enzyme, that's for sure. Okay, now what happens next? When I look at this kind of a diagram, I need to first kind of see the big picture. And what I see when I look at this diagram is first it's a circle. I start with something that I end with. It's a set of reactions. And what else do I see? I see a lot of NADH being made. Oh, well, one of these is FADH2. FADH2 is another shuttler of high energy electrons. FAD can be reduced to FADH2. It can pick up high energy electrons. Think of it as a smaller dump truck. It doesn't have quite as much energy as NADH, but yeah, we're making these reduced electron shuttlers. What else is happening in this citric acid cycle? Hey, more carbon dioxide is being released along the way. In two of the steps, carbon dioxide is being released as a waste product. In one of the steps down here, we're also seeing ATP being made by essentially the transfer of a phosphate group from molecule to molecule, and finally this phosphate group arrives at ATP. Wow. What's happening in the citric acid cycle is our molecule acetyl-CoA is being completely oxidized, and the resulting waste products, carbon dioxide, are released. But as it's oxidized, other molecules are being reduced, being made, that carry high energy electrons around. Those are NADH and FADH2. Okay? So let's see if we can fill in our stars here. There are a series of redox reactions in which NAD plus and FAD are reduced. They are the ones picking up those high energy electrons. The substrates are oxidized, right? Carbon dioxide molecules are lost. ATP is produced by substrate level phosphorylation. And at the end of this all, we started with oxaloacetate, we end with oxaloacetate. Those are the most important things to remember about the citric acid cycle. We're taking and tapping into all the energy in pyruvate, releasing the waste product of carbon dioxide, making a lot of reduced electron carriers, like NADH and FADH2. Yay, that's great. Shuttlers of high energy electrons. Let's briefly look at the cycle, and then we will kind of tally up what's happened again. So acetyl-CoA reacts with oxaloacetate in step 1 to form a 6-carbon molecule. Oxaloacetate has 4 carbons, acetyl-CoA has 2, you mix them together, you got 6. In step 2, it just looks like things are rearranged a little bit. Step 3 is definitely a redox reaction. And I know that because I see NAD plus picking up electrons, being reduced. Isocitrate must be being oxidized. And I bet the enzyme required for this step is going to be a dehydrogenase enzyme of some type, because they're seen and used to catalyze these kinds of redox reactions. It also looks like we're losing some carbon dioxide, right? That carbon dioxide is released, and now we have alpha ketoglutarate, which only should have 5 carbons. Step 4 looks a lot like our prep step. Carbon dioxide is released. We oxidize alpha ketoglutarate into succinyl-CoA. NAD plus is reduced to make more NADH. This coenzyme A gets reattached temporarily here. So now we've got succinyl-CoA at the end of step 4. By the way, this reaction probably required another dehydrogenase enzyme. It's a redox step. In step 5, it looks kind of complicated, but basically in step 5, succinyl-CoA is removed. It's replaced briefly by an inorganic phosphate molecule. But then that phosphate is donated via a couple of steps to make ATP. So the phosphate's kind of jumping around, and it's used to make ATP by substrate level phosphorylation. So we've made some ATP too. That's great. Now we have a molecule that has 4 carbons in it. It has 4 carbons. It's called succinate. Step 6 is another redox reaction. In this reaction, succinate is oxidized to fumarate. It loses hydrogens and their associated electrons. It loses electrons. FAD picks up those electrons and two protons and becomes FADH2. It's reduced to FADH2. Yay! We've made another little shuttler of high-energy electrons. Fumarate in step 7 picks up some water to become malate. And step 8 is another redox reaction. Malate is oxidized to oxaloacetate. NAD plus picks up those electrons and is reduced to NADH. I bet you know what type of enzyme is needed for this step 8 reaction to happen. Dehydrogenase. Okay. Whew. This is messy. I made a mess of it. I like all these names. We don't have to memorize them, but wouldn't it be fun to have, a fish tank full of fish with these names like fumarate and succinate and alpha-ketoglutarate? I don't know. Anyway, I think we have to remind ourselves that all of these steps, and even the pyruvate oxidation step, happens twice. If we're following one molecule of glucose going through aerobic cellular respiration, we ended up with 2 pyruvates at the end of glycolysis. And then both pyruvates are going to go through the pyruvate oxidation step and the citric acid cycle. So we really have to kind of double the products that are in this picture. Because if we're following one molecule of glucose, we have 2 pyruvates being moved through pyruvate oxidation and the citric acid cycle. Okay. Let's do some summarizing, actually, of the products that we've gotten. So we're going to summarize how many products we've made during pyruvate oxidation and the citric acid cycle, okay, both of those. And first we're going to think about the gain per pyruvate molecule, but we're really kind of interested in the gain per 2 pyruvate molecules if we're following one molecule of glucose going through aerobic cellular respiration. So we're going to kind of double everything and think about this bottom part of the chart. That's really the most important for our kind of purposes. Okay, so how much ATP did we see being produced during pyruvate oxidation and the citric acid cycle? Well, you might need to go back a couple of slides and take a look. No ATPs in this step, but I see an ATP by substrate level phosphorylation down here. So I'm going to say 1 per pyruvate molecule or 2 ATP. All right, we made some ATP. That's great. How about NADHs? 1 NADH from pyruvate oxidation, and then 1, 2, 3 from the citric acid cycle. So a total of 4 NADHs. Wow, that's a lot. That means if we're following 2 pyruvate molecules, we get 8 NADHs. That's a lot. Okay, those are those high-energy electron shuttlers. FADH2, I only saw that once, okay? We made 1 molecule of it per pyruvate, or 2 molecules of FADH2 if we're following 2 pyruvates. And how much carbon dioxide was released along the way? Well, let's go back. Here's 1 molecule of carbon dioxide in the pyruvate oxidation step. and then one more, and then one more. I see 3 molecules of carbon dioxide being released when 1 molecule of pyruvate is completely oxidized. That doubles to 6 CO2 for 2 molecules of pyruvate. Carbons are conserved in this process. Remember, we started with glucose. How many molecules of carbon are in glucose? 6. And now we're releasing 6 carbon dioxides. The carbons are conserved. We have completely oxidized glucose now. And we've released the waste products, 6 CO2s released. All right? So we've made some good things. 2 ATP, 8 NADH, and 2 FADH2. We're ready to use the NADH and the FADH2 that we made in the next step of cellular respiration called oxidative phosphorylation. We'll see you there.